morning, everyone. It's my pleasure today to be with you at the I1 workshop and to present my PhD study for all of you. My study was about developing a biosensor based on natural biopolymer for early detection of folate receptor cancer biomarkers. And in the next study, we make development by using one of the conjugated polymers, which has intrinsic fluorescent and electrical conductivity, to examine if there is enhancement of the performance or not. The title was poly-3-hexylthiophene, which is the conjugated polymer, based bioelectronic interface using conducting nanofibers for the sensitive determination of folate receptor cancer biomarkers in human plasma. As we see, the first publication was published in the International Journal of Biosensors, and we published a review article about the conjugated polymers. This review was selected by the editors as a cover for issue 8 in Microorganism Journal. And the last study was after adding our conjugated polymer, poly-3-hexylthiophene, published in Analytical Letters. We prepared our polymer solution from sodium alginate and polyethylene oxide with our biorecognition elements, folic acid, and aqueous solution. After that, we added the B3HT and make a stirring until the color changed to purple, which confirmed the formation of nanoparticles due to the non-solvent aqueous medium. And we make electrospinning technique to obtain our nanofibers. Then we make deposition of the folate of nanofibers solution on the surface of working electrode using screen-printed electrodes. Then we incubated with the folate receptors solution in human plasma, and we make detection using EIS technique or electrochemical impedance spectroscopy. Here we make characterization using SEM or scanning electron microscope. When we optimized the ratio of B3HT, it was above 1%. We obtained the bead morphology, which is not recommended for our biosensor. So we started to reduce the amount to less than 1%. We obtained a very homogeneous structure with the smooth surface area and the small diameter around 84 nanometer, which confirmed a large surface area. Here we examine Here we make characterization using FTIR technique. We obtain the distinctive peaks for each polymer. For example, the broadband between 3,000 3,500 nanometer for the hydroxyl groups of sodium alginate, and the bands at 2,800 for the hexyl chains of B3HT. And after we added the folic acid, there is a decrease in all the bands. This may be related to the hydrogen bonds between folic acid and hydroxyl groups and the hexyl chains. And we continue characterization using electrochemical characterization using cyclic voltammetry and EIS technique. As we see, there is increase in the current peak from the blue dashed curve for screen-printed electrode without any modification to our conducting nanofibers layer with red color, and increase more and more after conjugation with folic acid. This due to the increase the ionic conductivity due to folic acid and the nanofiber structure with the large surface area and the porous structure, which allow the transfer for the charge. And also the redox activity and the electronic conductivity of B3HT. Here I want to point to this electrochemical characterization in solution containing redox marker as ferroferricyanide, and in another one without any redox marker, depending only on our conjugated polymer. As we see the same behavior, there is increase in all the signals of cyclic voltammetry, which confirmed that we can depend on the redox activity alone without any redox marker to follow the interfacial change on our electrode. The same behavior in the impedance, as we know, if the current increase, the resistance to charge transfer decrease. So the impedance decreased gradually from the screen-printed electrode and after modification with conducting nanofibers, and decreased more and more after conjugation with folic acid in both solutions. Here we examine the effect of different scan rates in the range of 10 to 100 millivolt per second on the intensity of peak current in both layers for the conducting nanofibers and after conjugation with folic acid. And the same, we obtained increase in the peak current, and we make fitting and extract the value of IB, which is peak current, against square root of scan rate. We obtained a linear relationship. This linear relationship confirmed that our electrochemical process based on diffusion technique, which is very, very recommended for our biosensor, and confirm enhancement of sensitivity. From the slope of this curve, we obtained this equation, which is Randles-Civic equation, to calculate a very important parameter, which is A, electroactive surface area, which means electroactive species allow the reaction between folic acid and folate receptors. And the remaining are constant, the number of electrons and the diffusion coefficient, the concentration of redox molecules, and we obtained increased in this value. Also, from Volmer equation, we calculate the constant for electron transfer. This parameter also is very important because it reflects the speed or kinetic of electron transfer in the electrolyte solution. And of course, if there is increase in both these parameters, the electroactive surface area and the electron constant transfer, this confirmed enhancement of the sensitivity and the amplifying the signal of our biosensor. Here we investigate the detection behavior of our biosensor for folate receptor and trace HCl buffer. As we see, there is increase in the diameter of semicircuit of Nyquist plot. This increase in the diameter increased gradually with increasing the concentration of folate receptors. And this confirmed the sensitivity of detection of folate receptor even in the buffer solution. It was around 10 nanomolar, the concentration of our protein. Here, this histogram shows the difference between the performance of our biosensor with B3HT and without B3HT. As we see, there is a significant difference, which confirms the additive effect of our conjugated polymer. Here, we examined the selectivity using other interfering plasma protein, which is human serum albumin, and there is a significant difference, which is confirmed the higher selectivity. Also, we stored our biosensor for 2 months at 4 degrees C to see if there's stability or not, and we obtained nearly the same signal. We make fitting for all the data to extract the value of RCT, which is the resistance of charge transfer. It was nearly almost the same value. These results encouraged us to investigate our biosensor and human serum plasma. We prepared our human serum plasma and spiked it with our folate receptor protein in a wide linear range from 1 picomolar to 100 nanomolar. As we see, there is increase in the circuit diameter with increasing the concentration of folate receptors. We make fitting for all the data and extract the value of RCT and calculate the variation of RCT to plot against the logarithmic concentration of the protein to obtain from the calibration curve our limit of detection. As we see from the calibration curve, there is 2 slopes. This may be due to the many binding vacancies in the structure of nanofibers allowed the reaction between folic acid and folate receptor in the low concentration, and the remaining part of folic acid in many binding sites in the nanofiber structure allow the reaction at higher concentration. Here I want to point to that the limit of detection was about 0.1 picomolar. The lowest limit of detection related to the published articles for the same biomarker, and with enhancement 25-fold increasement related to our last publication. We continued measuring the calibration curve, but here we generate our calibration curve against the concentration only, not logarithmic, using Langmuir equation model to obtain the apparent affinity constant between folic acid and folate receptor. It was around 4.4, nearly the same value obtained from previous study. We investigate also the reproducibility of our biosensor. It's a very important parameter because simply if any biosensor not reproducible, it can't be applicable. We carried out the experiment in the same condition under the same factors make the same optimization for all the steps, and we obtained nearly the same behavior in the freshly prepared 3 electrodes as we see. And we make fitting for all the data to extract the RCT value and calculate the variation. We also obtained the same limit of detection. And finally, we examined the biocompatibility of our biosensor by incubation with the MCF7 pressed cancer cell lines, and for the 3 layers, the biopolymer nanofibers, and after conjugation with B3-HT, and after adding folic acid. We obtained not significant cytotoxic effect against cell lines. The bioavailability was around 90%. Exception the 3rd layer after adding folic acid, maybe there is little significant difference. We think this result is normal because folic acid mediates the pathway for enhance the cellular uptake by cancer cells due to overexpression of folate receptors on the surface of cancer cells, which also this result opened the way to investigate this biosensor more in theranostic application, which is mean the diagnosis of cancer cells at the same time treatment of cancer cells using target drug delivery. Here I want to announce it was my pleasure a few days ago in Egypt. I won from my PhD study the 3rd place in prize in entrepreneurship category in the biggest biotechnology competition in Egypt called the SOL, or Science Operational Leaders in Egypt 2025. Finally, I would like to thank my supervisor and my role model, Professor Hafsa Kori Yousefi. She's the leader of Biosensor Group and Biochefs for Analytical Device at Paris Saclay University, and all my friends and our team. Thank you.
Thank you, Dr. Noha. We will take one burning question. Yes, please.
Thank you for the presentation. I just want to know which redox couple you have used for the sensing.
Ferroferricyanide.
Okay, and you are using carbon screen printed electrodes, right?
Sorry, what?
Screen printed electrode material is carbon, na, which you are using?
Yes.
Okay, thank you.
You are welcome.
Thank you very much.